Assalamu alaikum dear students. Today's topic is DNA replication. DNA replication. Formation or synthesis of DNA from DNA is called as DNA replication. This is DNA. This DNA forms DNA. Then RNA and finally proteins. Formation or synthesis of DNA from DNA is DNA replication. Formation or synthesis of RNA from DNA is called as transcription, and formation of proteins from DNA through the synthesis of RNA is called translation. First of all, we will discuss DNA replication. This is cell. This cell contains nucleus, and this nucleus contains DNA. DNA replication occurs in the S phase of cell cycle. S phase of cell cycle. There are three stages in the interphase, namely G1, S, and G2 phase. In this phase, DNA replication occurs. Since we know that DNA is double-stranded, it is one strand, and it is another. It is 3 prime. It is 5 prime. It is 5. It is 3. DNA replication starts or initiates at a specific point is called as origin of replication. In bacteria, there is a single origin of replication, but in case of eukaryotes, there are multiple origin of replications. And this origin of replication is also called as ORIC. ORIC. When DNA replicates, first of all, its two strands are separated by the enzyme called as helicase. This helicase is also called as DNA B protein. This helicase attacks at AT rich region as compared to GC rich region, because there is double bond between A and T as compared to G and C. There is triple bond. So ORIC is rich in AT rich region. First of all, DNA helicase, it separates the two strands. Then comes the enzyme called as SSB. Single strand binding protein. It keeps the two strands separated. That means it prevents re-annealing, like this. Now there are two strands, upper and this lower one. This one is called as leading strand, and this one is called as lagging strand. In this leading strand, DNA replication occurs normally because DNA polymerase third works in 3 to 5 dash direction, and there occurs 3 dash OH nucleophilic attack. Then there occurs polymerization reaction, and synthesis of this strand occurs. Now there is this strand, lagging strand. This strand presents the problems because DNA polymerase works only in 3 to 5 dash direction. It occurs in pieces or fragments called as Okazaki fragments. Okazaki fragments. First of all, this is now lagging strand. These are now Okazaki fragments. First of all, RNA primase lays down RNA primer. This primer provides 3 hydroxyl group, that means OH group. Now comes the enzyme DNA ligase, which joins these Okazaki fragments. That means there is a formation of phosphodiester bond performed by this enzyme between 3-OH fragment and 5- phosphate group on other side. It is joined by DNA ligase. There occurs phosphodiester bond. Ultimately, this RNA primer, it is removed by enzyme RNase H. But still there remains the last nucleotide like this one. It is not removed by this RNase H, it is removed by an enzyme called as PEN, first or flap endonuclease first. But in case of eukaryotes, we will not find their DNA pol first, second and third. Instead there are the enzymes like this. In case of eukaryotes, there is DNA polymerase alpha, DNA polymerase beta, gamma, delta and epsilon. This one, the function of this is priming. The function of this one is base excision repair. The function of this one is mitochondrial DNA replication. The function of this one is synthesis of lagging strand. And the function of polymerase epsilon is synthesis of leading strand. This is priming, it is base excision repair. If the DNA base gets damaged, it is repaired by DNA polymerase beta. Mitochondrial replication is performed by this gamma. Delta is performs lagging strand synthesis and epsilon performs leading strand synthesis. This was a brief account about DNA replication. Thank you.